vector, or sort of a carrier of that therapeutic nucleic acid. And in one case, you directly inject that carrier into the patient where it will deliver the therapeutic gene to the proper cell type. But in another way to sort of carry out gene therapy is to actually remove the patient's cells, treat them with this viral vector, and then reinsert those altered cells into the patient. Now I want to emphasize that the major barrier to gene therapy, having commercial success is really developing an effective carrier that can deliver these nucleic acids to the proper cell type. And so when it comes to the choice of a carrier, there's really two main classes. And this pie graph actually shows the distribution of different carrier types in human gene therapy clinical trials as of 2015. And so what you'll notice is that 60 to 70% of vectors in gene therapy clinical trials are actually viral based. Now, although they're much more popular than synthetic vectors due to their efficacy, they actually suffer from numerous drawbacks, which include safety concerns such as improper insertion of the gene into the patient's genome, which can result in side effects, as well as eliciting an immune response from the patient, because the patient's immune system will actually recognize the protein from the viral capsid. Now, that's not to say there hasn't been success with viral vectors. Glybera is actually the first commercially approved gene therapy, which is slated for release in 2016. But its projected cost for a single dose, a single injection, is $1.6 million. So it will be the most expensive medicine actually available on the market. So another sort of benefit to using synthetic vectors is that their cost and their scale of production is much easier. And so in this pie graph, what I've highlighted here is that 5% of human gene therapy clinical trials actually use lipofection or synthetic vectors. Now, when I say a synthetic vector or lipofection, I'm talking about macromolecules that can actually be synthesized in the lab using a chemistry. And a natural choice for a synthetic vector is actually a liposome or a lipid-based system. Now, as of 2012, there are 12 FDA-approved drugs that actually use liposomal delivery systems. So it's a pretty natural choice to go ahead and see if liposomes can also deliver therapeutic nucleic acids as well. Now, it was initially discovered in 1987 that the complexation of cationic liposome DNA improved the internalization of that DNA into mammalian cells in vitro. And numerous work has been done in the decades since then. Even today, Alnylam Pharmaceuticals is actually passing phase 2 clinical trials with a cationic lipid-based treatment for transthyretin amyloidosis, which delivers siRNA, another type of therapeutic nucleic acid. So there's a lot of work that's been done, but there's still really a long way to go for gene therapy, particularly gene therapy using synthetic vectors, to really reach its full potential. And that's what I'm going to talk about today, sort of the work I've done to help really push this whole field forward, even if just a little bit. And so initially, my talk is really going to have 3 parts. And in the first part, I'm going to talk about previous work done in the Safina group that looked at the structure as well as the mechanism of action of these lipid DNA complexes. And during this part of my talk, I really want to highlight some of the different techniques I'm going to use to investigate these particles. In the next 2 parts of my talks, I'm really going to go into my work and what I've done. And then the first half, I'm going to talk about novel structures I've discovered upon adding PEG lipid to these lipid DNA complexes. And that work is going to be focused, really driven by cryo-EM. And in the second part of my talk, I want to show how fluorescence microscopy can provide mechanistic insight really into how these lipid DNA particles deliver therapeutic nucleic acids at the single cell level, and really what the major bottlenecks to delivery are. And so the first question I want to answer is, what is the actual structure of cationic lipid DNA complexes? And so in this schematic, what's shown here was the initial hypothesis, which is that the DNA in cationic liposomes form sort of a bead on a string structure because of their opposite charge. You have electrostatic attraction between the 2. But in 1997, it was shown through small angle X-ray scattering that mixtures of cationic lipids and DNA actually form well-ordered phases, okay, well-ordered condensed phases. And here's an example of such a phase. So what's even more interesting is that the structure of these phases actually depends on the chemistry of the lipid and its shape. And so if we use a lipid like DOPC, which has a cylindrical shape, and mix it with cationic lipids as well as DNA, they will self-assemble into what's called the lamellar alpha phase, where we have alternating sheets of lipid bilayer And in between these lipid bilayer sheets, we have a DNA forming actually a two-dimensional pneumatic. Okay. Now, if we use another neutral lipid like DOPE, which has this inverted cone shape, we'll see that the self-assembly actually will form, the structure will look like DNA coated in lipids packed on a two-dimensional hexagonal lattice. Right? So again, you have lipids that have sort of different intrinsic shapes. They prefer different surfaces of different spontaneous curvature, and they drive the self-assembly of lipid DNA complexes into either one of these structures. Now what's interesting is that alongside the shape, the bending rigidity or sort of the floppiness of the membrane can also play a role in what sort of structure is preferred. And so by adding a single-tailed surfactant, which makes this membrane less rigid, you can induce a phase transition from the lamellar alpha phase into this hexagonal 2 phase. And that's because in the H2 phase, you really have complete neutralization of the negative charges along the DNA backbone. Now, the structure of these lipid DNA complexes is fascinating from a soft matter point of view. But ultimately, we want to design and optimize lipid DNA complexes for therapeutic delivery. And so we need means or methods, ways to study how these lipid DNA complexes actually interact with cells. So one technique that I'm going to frequently refer to throughout my talk is what I call a transfection efficiency assay. And with transfection efficiency, we have a reporter gene here. This is PGL3, so it will code for a protein which can produce photons either through bioluminescence or fluorescence. We'll complex that DNA with the lipids, forming these lipid DNA complexes. And then we will actually add these complexes to cultured cells, wait for the allotted amount of time, and then effectively count the number of photons that are produced. And this gives us a way to really understand how efficient different lipids and different formulations are at bringing nucleic acids into cells.
But the fluorescence molecules are on the surface, or?
No, so this is coding for the fluorescent molecule.
Oh, it codes. I see.
Yeah, it codes
Genetically
for the
coded.
cell.
So
Yeah, the cell will produce it.
it's a bit produces itself. Thanks.
Yeah. And so this is what the results of such a measurement would look like. And on the y-axis, you have relative light units, so it's a measure of photon count. And what I've plotted here, what was done about 10 years ago, is that different cationic lipids have a different optimal transfection efficiency based at different mole fractions. Okay? So each one of these colored symbols represents a different lipid. As we vary the mole fraction cationic lipid by adding more neutral lipid, we'll see that sort of the optimal is happening at a different spot. But what was interesting and very unexpected is that if you rescale this x-axis to membrane charge density, which is a function of the lipid valency as well as head group area, you'll see that all these points actually fall on a universal curve. So there really is sort of universal behavior happening in terms of how these nucleic acids are delivered into cells. And what you'll notice is that on the universal curve, there's really 3 distinct regimes. And to understand what's happening in these 3 regimes, the Aisha Ahmed, a former graduate student in Sir's group, used fluorescence microscopy. And so these are fluorescent micrographs of cells that have been incubated with dual fluorescently labeled lipid DNA complexes. So everywhere you see red is an indication that there's lipid, and everywhere you see green means there's DNA. And when those 2 overlap and form a complex, you'll see a yellow spot. And so at low membrane charge densities, we see colocalization of lipid DNA, indicating there are intact complexes inside the cells. And at high membrane charge density, we see the free DNA signal inside the cell green. And so piecing this, putting this information together with the transfection efficiency assay, we can really put together a mechanism for how these lipid DNA complexes deliver nucleic acids and what the major barrier is. So initially, all of these, regardless of how high or low the membrane charge density is, they're all net positive, so they stick to the outside of negative cells through electrostatics. They undergo a process called endocytosis, which is how cells engulf material from the outside. They will be trafficked in a vesicle called an endosome. And at low membrane charge density, they remain trapped in these endosomes where they'll eventually be degraded. This is why we see co-localized lipid DNA signal in those fluorescent micrographs. But at high membrane charge density, they'll actually fuse with the endosomal membrane and release the complex as well as DNA into the cytoplasm. And this is why we see the free DNA signal in those fluorescent micrographs. Now, you'll remember in the universal curve, it sort of had a bell shape. So at even higher membrane charge density, the lipid DNA complex will escape, but DNA will not disassociate due to the high amount of electrostatic attraction between the lipid DNA. So this is really just an example of how fluorescent microscopy and transfection efficiency assays can sort of provide insight into how these lipid DNA complexes sort of deliver genes and why they don't work in certain cases, such as regime 1 here and regime 3. Now, developing lipid DNA complexes for use in vitro has been done, again, as I mentioned earlier, for decades now. And there are numerous commercially available reagents for doing nucleic acid delivery in vitro. In fact, one of the lipids that was custom developed in the Safina group is now available from Avanti Polar Lipids for doing siRNA delivery in vitro. Interestingly enough, some work I did with Xiaoling Chen, a postdoc and Kaiyuer, a project scientist, resulted in the discovery of a lipid formulation that is very efficient at delivering nucleic acids in the presence of serum. And Avanti, the company selling this, is actually going to sell this lipid formulation as well. So, doing transfection in vitro, I don't want to say it's solved, but there are numerous options commercially to do it. And really the next barrier for synthetic vectors is to show that same type of efficacy in vivo, or in the living organs. And so the problem is when liposomes or lipid DNA complexes are injected into living organisms, they're rapidly cleared from circulation by the immune system. Okay? And this happens by being marked by a protein called opsin, which is an indication of macrophages to remove the foreign body from circulation. So one technique to extend the circulation time of these drug or nucleic acid carriers is to graft their surface with a polymer that prevents the binding of these opsin proteins. And a great choice for a polymer is actually polyethylene glycol, or PEG. Okay? PEG is already FDA approved. It can be grafted to the head group of a lipid, and that allows you to easily cover the surface of these liposomes or lipid DNA complexes with this PEG molecule. Now, unfortunately, there's sort of a dilemma here, because although PEG was shown in vivo to increase the circulation time of liposomes as well as individual proteins, it significantly reduces the efficacy of these lipid DNA complexes in vitro. So it helps overcome a barrier in vivo, but it's creating an entirely different barrier in vitro. And this is really where my work comes in. So I wanted to study, first of all, what structures form when we add these PEG lipids to lipid DNA complexes. How does the structure change? And I also wanted to see what sort of mechanistic insight can I gain from doing fluorescence microscopy with these PEG-related nanoparticles. And so here is a cryo-EM micrograph of a lipid DNA complex that does not contain PEG. And you'll see that it forms a large aggregate with a striation pattern, which is actually indicative that it's in that lamellar phase I showed on the earlier slide. But when we add PEG lipids to these lipid DNA nanoparticles, we form stable sub-100 nanometer nanoparticles that do not fuse even at very high local concentrations. So they're resistant to aggregation. Right? And this is because, again, the PEG forms a steric barrier around each one of these nanoparticles. And so, it's unsurprising that at equilibrium, these lipid DNA nanoparticles coexist with liposomes. And that's because they're formed at a charge ratio, or a lipid to DNA ratio of 3, meaning there are 3 cationic charges for every anionic charge in the DNA. And this is done to ensure that these lipid DNA nanoparticles have a net positive charge. And what happens is the excess lipid forms these liposomes shown here. And so what was sort of unexpected was that, and alongside these nanoparticles and liposomes, we also see these small branched structures. Now, cryo-EM has relatively low contrast, so in an image like this, you can't really measure sort of the diameter of these structures or really understand what they are. And so we're going to use basically a sample prep trick to pack them at a very high density. And so cryo-EM works by having a substrate containing holes shown here. You add your solution to it, and you remove the excess water from that solution. And that forms this thin film right here of water containing your sample. Now, the thickness of the water varies, and the center of the water film can go down to as low as 10 nanometers. And when this happens, it will exclude larger objects from the center of the hole. Okay? So when we find a hole that sort of has the right geometry, this is what we see. This is basically the outer edge of one of these carbon holes. And you can see near the outer edge are all the larger objects, like the lipid DNA nanoparticles, as well as the liposomes. But in the center of the hole, we have a very high local concentration of these thread-like structures. And you can see that the spacing between them is very uniform. They're very well ordered. And with an image like this, we can actually really measure exactly what are these characteristic length scales that we're seeing with these structures. And so to do that, we coming from an X-ray group, using a Fourier transform is a very natural choice. So the Fourier transform of this box region is shown here. And what you'll see are there are two bright rings surrounded by this sort of dark band. And if we do an azimuthal integral around this Fourier transform, this is basically the intensity profile. And what you'll see are there are these two main peaks here related to these two bright bands, as well as this change in slope right here. These two sharp peaks are basically a result of what's called the structure factor. And so their location in reciprocal space is related to the distance between these structures, the spacing in between these micelles. Now this change in slope is really due to what's called the form factor. So its location in reciprocal space can give us the actual thickness of those micelles. And what you'll notice is that the spacing between them is about 14 nanometers, and the thickness of each micelle is about 3.7 nanometers. And with these length scales, we can really put together what these thread-like micelles are composed of. And they're actually cylindrical micelles containing PEG lipid on the outside. This 3.7 nanometer thickness of the micelle is in perfect agreement with bilayer, with the thickness of a bilayer that's been measured with X-ray scattering as well as surface force apparatus. And this 14 nanometer spacing in between them is consistent with having two surfaces separated by PEG and a brush conformation. And so this was one sort of unexpected structure, these thread-like micelles that we found when we mixed pegylated lipids and cationic lipids in DNA. And what we really wanted to do was understand.
Sorry, just a question. So the DNA, is the DNA in here? Or
no,
not?
the DNA is not in
this
here.
is just,
Yeah, exactly. Because if the DNA was in here, you'd expect it to be a little bit thicker.
Yeah, actually.
Yeah.
Okay.
And so I'll explain sort of what drives their formation and why. Again, it'll sort of tell you where the DNA is in this picture. And so, as I showed earlier, the lamellar complex has this structure where you have alternating bilayers, two-dimensionally ordered DNA in between. And what was shown is that the addition of neutral lipid increases the spacing between DNA by basically increasing the total amount of available bilayer area. And a later discovery showed that the addition of PEG lipid reduces the DNA spacing by inducing a depletion attraction force. And what I found is that when you already have DNA with a very small spacing in between, and you add PEG lipid to something like this, the lipid DNA complex will expel PEG lipid as well as neutral and cationic lipids into these thread-like micelles. So we have PEG lipids and DNA competing for a finite amount of bilayer area. And they sort of resolve this frustration by coexisting with these two structures at equilibrium. Okay, now we can sort of infer that the composition of these thread-like micelles also contains DOPC and DOTAP along with PEG-2000 lipid, because PEG-2000 lipid by itself would prefer a surface of positive spontaneous curvature, meaning a spherical micelle. And a cylindrical micelle has this positive spontaneous curvature around the diameter, around the circumference of it, but along the thread-like micelle, it actually has zero spontaneous curvature. So it's actually indirect evidence that these thread-like micelles are really a two-component system, a lipid that prefers positive curvature as well as one that prefers zero. Now, another structure that we sort of discovered was initially sort of motivated by our DLS finding is these DNA tethered nanoparticles. So what I'm showing here is the size of these nanoparticles measured with light scattering as a function of this charge ratio, this ratio of lipid to DNA. You'll notice in the case of a plasmid DNA, the size, the sort of the shape of the size function here is really consistent with electrostatics, where near the isoelectric point where you have charge neutralization, there's very little electrostatic self-repulsion, so you form large nanoparticles. As you move away from the isoelectric point, you see that they actually shrink due to the electrostatic self-repulsion. But with long DNA, the sort of unexpected finding was that as you continue to reduce the charge ratio, it continues to grow in size. And so we were initially puzzled by what's happening here, but we used imaging to actually understand what goes on when we use a lot of this long lambda DNA. And what happens is that you're forming a tether between individual nanoparticles. So here's a lipid DNA nanoparticle. This is the excess DNA in solution because we're at a very high DNA to lipid ratio. And what you'll notice is that these individual nanoparticles are actually tethered to this nanoparticle right here. Now, this is interesting because this could really potentially be a new class of synthetic vectors where you can deliver large genes through tight spaces by having nanoparticles that are in close proximity, sharing one strand of DNA, and capable to basically reorganize. Now, again, I've shown you these interesting structures that form, but ultimately we want to optimize these lipid DNA nanoparticles for use in vivo as well as in vitro. And so to do that, we're really going to turn to fluorescence microscopy to understand what are the bottlenecks to efficient nucleic acid delivery. So as I mentioned on an earlier slide, if we look at the transfection efficiency versus the amount of PEG lipid mole percent, we see that there's a massive drop at 5 mole percent. Okay? This drop in transfection efficiency coincides with the transition of these PEG polymers from a mushroom configuration to a brush configuration. Now, this drop in transfection efficiency had been reported in the literature, but there were sort of conflicting reports as to what's causing it, really. And the two main models were that it reduces cellular internalization. You'll remember that there's an electrostatic mediated attachment of these nanoparticles to cells. So by having this PEG layer, maybe it's preventing it. And the other model was that they could not escape endosomes, because again, this is requiring the nanoparticle to come into close proximity with a membrane, with one of the cell's membranes. And so to really understand this, we turned to Kai at UR, who's a project scientist in Cirrus group, and he designed custom PEG lipids that would really allow us to test those two hypotheses. So this is sort of our control case right here, just a pegylated lipid DNA nanoparticle. And here we have a pegylated lipid DNA nanoparticle where we have a targeting peptide at its distal end. And here we have a pH-sensitive nanoparticle that can shed this PEG layer at low pH. The idea being that if cellular internalization was really the main barrier, putting this peptide at the distal end of it should recover that internalization. We should see high transfection efficiency. And if endosomal escape was the main barrier, then having this pH-sensitive PEG that's capable of cleavage in the low pH environment of a late endosome should recover transfection efficiency. Now, the targeting peptide that we chose was actually RGD. So RGD is a 3 amino acid sequence that's been shown to target a membrane-bound receptor called integrin, which can be overexpressed on cancer cells. And today, I'm going to show you our results with linear RGD. Okay? Kai has also synthesized these cyclic RGDs, which have a higher binding affinity, but we've actually learned a lot about the internalization and the endosomal escape of these properties just with linear RGD. And so here are fluorescent micrographs that have been incubated with pegylated lipid DNA nanoparticles. And you'll notice that at low membrane charge density, we don't in fact actually see any nanoparticles inside the cell at early time points. And at later time points, we see one single nanoparticle that's actually internalized. But when we add RGD to the distal end of the PEG, we recover binding to the plasma membrane at early time points. And at later time points, we actually see an abundance of these particles inside the cells. Now, I want to also highlight that because these are pegylated, they are resolution-limited spots. They're actually resolution-limited spots. So these individual spots here could be one or multiple nanoparticles inside an endocytic vesicle. Now, we wanted, outside of qualitatively showing that RGD works, we wanted a way to quantify this. And to quantify something like this, we need to be able to average results over numerous cells. So I've developed here really a software routine that allows us to measure the intracellular location of these individual nanoparticles. We use the nuclear membrane as a reference point, so now we can average these results over numerous cells. And this is sort of what the output of such a routine would look like. So what we're plotting here is the number of fluorescent spots versus distance to the nuclear membrane. We see that in the case when it's just pegylated, we see practically nothing is being internalized inside cells. And when we add this RGD peptide to the distal end of PEG, we see that we recover internalization as well as accumulation of these nanoparticles near the nuclear membrane or near the nucleus. And so what was interesting is that now if we vary this membrane charge density, which we found to be a very important, which was shown to be a very important parameter for complexes lacking pegylated, we see that there's really two means for these nanoparticles to enter cells. In one case, you have non-specific electrostatic adhesion of these nanoparticles to the plasma membrane, because you can see that it's actually the number going inside is increasing with membrane charge density. And in another case, we see that RGD is mediating the uptake, presumably through binding to integrin. And here, we don't see a dependence on membrane charge density. The total number of cells that go inside is completely controlled basically, but we see a slight increase with membrane charge density.
That's why I'm just confused. I mean, you're looking at how many of these get inside the cell.
Yeah.
But then you're plotting versus distance to the nucleus of the cell. So how are those related? I mean,
so the integral of each one of these curves basically gives you the total number of the cell.
So why do you plot it this way? I mean, I know you want to get them to the nucleus.
That's a fair point. So this showed us that they're accumulating next to the nucleus,
which is what you
which
want.
means they're being trafficked, right?
Because
if they're free in the cytoplasm, you'd expect a uniform distribution.
So how big is the actual cell?
So this is about 30 microns. You know, each cell from about the center to the outside of about 0 to 50 microns.
The longest distance. The
longest distance, yeah. But of course, now there's a lot of variability in cell size and shape. That's really why it gets noisier as we get to the end here. And so this finding here that the membrane charge density could actually control internalization is really consistent by considering the interaction between one of these nanoparticles and an anionically charged membrane, shown here. So, you know, this particle and this oppositely charged surface interact through all these terms, which include electrostatics, van der Waals, a hydration repulsion term, and a steric repulsion term due to the PEG. And if we plot the free energy versus distance between a nanoparticle and the surface, we see that at low membrane charge density, there's no minimum in the free energy. But at high membrane charge density, a minima forms, indicating that this can actually adhere to this. Okay? So what we found here is consistent with this model, and this model really gives us a way to predict whether lipid nanoparticles that have different brush grafting densities, different charges, and different brush lengths would stick or not stick non-specifically to cells. Now, if we look at the transfection efficiency, we see that with no functionalization, we're on the left side of the universal curve, so we see an increase with membrane charge density. When we add PEG lipid, we see a massive decrease in transfection efficiency. Note that this is a log scale. And when we add RGD, we only see partial recovery. And the reason we see partial recovery is because the steric repulsion of the PEG-RGD motifs inhibits endosomal escape. So although the RGD recovers the internalization of these particles, we still have to escape the endosome, which is why we don't see complete transfection efficiency recovery. Now I want you to note that again, high membrane charge density, like the unfunctionalized case, does promote endosomal escape, right? We do see an increase as we increase membrane charge density. But we can't quite get full recovery. And so this was sort of the model here for what happens when you pegylate an RGD tag, which is, you know, with RGD tag nanoparticles, you have internalization, but then you have endosomal escape, and you never really get to, you never really put together a nanoparticle that's capable of releasing DNA as effectively as the nanoparticles lack competitively. Now, again, the membrane charge density does help the escape, but we looked at another means, again, using one of Kai's custom synthesized lipids to promote endosomal escape, and that's using one of these pH sensitive lipids. So here we have an acyl hydrazone bond that's actually prone to hydrolysis and cleavage at low pH. And the model is that as this endosome matures and lowers its pH, it will shed this PEG polymer from the surface, and this fusion step will happen. And so performing the same types of analysis, this is what we found. So here we see, this is actually the result of integrating each one of these curves, this inset. So again, it's the total number of particles for each cell. And the green is RGD, which we showed is higher than PEG in the earlier section. But what's interesting is that at early time points, the blue, which is this hydrolyzable PEG, is similar to PEG, and at later time points, we see a difference. Now, this could lead you to believe that the HPEG is internalizing more efficiently, But what's actually happening, if you look at the images, is that a single endosome will contain multiple nanoparticles. And as these hydrolyzable PEG nanoparticles escape the endosome, the distance between them becomes greater than the resolution limit of the microscope, and you can actually count more individual nanoparticles. So it's an indirect way to sort of see that these nanoparticles are escaping these endosomes. Now, what happened when we look at the transfection efficiency, again, we see only partial recovery between the case of no PEG and PEG. And that's because although these are very efficient at escaping endosomes, they don't internalize very effectively because they don't have the RG. So I've shown you really two barriers, right? There's the entering the cell as well as escaping the endosome, and I've shown you how two different custom lipids can sort of help nanoparticles do either one of those things. But ultimately, we need a better assay to really measure endosomal escape directly. Because the transfection efficiency assay is really a convolution of internalization, endosomal escape, entry into the nucleus, as well as transcription. And me and Circe wanted to develop a way to really directly measure how many nanoparticles escape these endosomes so that we could really design better formulations. And so to do that, we looked at this family of enzymes called Rab-GTPases. Rabs are responsible for the budding, trafficking, and fusion of membrane-bound organelle inside cells. And in humans, there's in fact over 70 different types. And so different Rabs mediate different steps of the endocytic process, shown here on the right side of the curve. But they're also involved in just intracellular transport, for example, here from the endoplasmic reticulum to the Golgi. And they're also involved in an exocytosis, or removal of cellular cargo into the extracellular space. But if we have fluorescent Rabs and we look for co-localization of these Rabs with nanoparticles, we can really track the intracellular pathway of these nanoparticles directly.
Are the Rabs in membranes of vesicles?
Yeah, exactly. The Rabs are in membranes, and I'm going to explain at the next slide how they work, what their job is. And so today, I'm going to focus on Rab5, which is a marker for the early endosomes, really the beginning of this endocytic process. And so this is the life cycle of Rab5 right here. It's a GTPase, which means it really has two states, one in the GTP-bound form and the other in the GDP. In the GTP-bound form, it's found on a membrane. And here, what happens is it will accumulate at the site of a clavering pig or macropinocytic ruffle, which will be internalized into the cell, basically pinched off from the plasma membrane. And Rab5's main purpose is to mediate the fusion between Rab5 containing vesicles, or what's called homotypic fusion. And so when these small vesicles containing Rab5 GTP begin to fuse, they form what's called an early endosome, or this early or sorting endosome. Now, after sufficient time, this GTP will be hydrolyzed to GDP, and the GDP Rab is actually returned to the plasma membrane in the cytosol. And so then the cycle continues. Right? So if we look at cells expressing Rab5 GFP, this is what we'll see. So now lipid and DNA are fluorescently labeled red and blue, and Rab5 is fluorescently labeled green. And everywhere we see these green spots, we're looking at early endosomes that are labeled with Rab5 GFP. And what I've highlighted with these arrows here are actually co-localized signals of Rab5 GFP and these fluorescently labeled nanoparticles. I've blown up this box region right here so you can sort of get a better example of this. This particle here, labeled I, is just a lipid DNA nanoparticle lacking co-localization with Rab5. And these two are actually co-localized with Rab5, implying that they're inside an early endosome. Now, what caught us off guard about this result is that you can actually see a lot of nanoparticles are not co-localized with Rab5, right? And so again, we've qualitatively demonstrated that these nanoparticles use a Rab5 pathway, but we really need a way to quantify how many are inside these early endosomes at a given time. Yeah?
Do you have a sense of, in one of these Rab5 spots, how many there are?
How many there are?
Right.
It's a little bit...
Is it a very small number, or is it a very large number?
It's a very small number. I'm going to show on a later slide the average size of one of these endosomes. And based off of that, you really can't fit that many nanoparticles in such a small space. But it's a little tricky to use the fluorescence because there is some polydispersity in these nanoparticles. But it is a relatively small number. Now, when you see a much brighter spot like what's shown here, this is probably, a collection of 4 to 5.
But they've got out of the endosome?
Yeah, so I'm going to explain why they're not co-localized and how we sort of figured that out. And so, we wanted to measure the number of them, so we had to develop our own co-localization assay that uses these fluorescent images. So here's the size. A typical endosome can be anywhere between 200 and 1,000 nanometers. And now RABs form domains on these endosomes, so they're not uniformly distributed across the entire surface. And a nanoparticle inside an endosome with a RAB domain could actually be up to 700 nanometers apart from that RAB domain, if we take 500 nanometers as an estimate for their diameter. And so, to develop this assay, I looked at pegylated and RGD tag nanoparticles. I varied the threshold for what I would consider co-localization, and I calculated the number of co-localized nanoparticles. And you see here, this green line represents 700 nanometers, which is a reasonable distance to say the nanoparticle is in an endosome. And you can also see that at 700 nanometers, you can detect differences between PEG and RGD. So this really sort of solidifies our understanding that we have an assay now where we can measure the number that are inside versus outside, and it makes sense with the spatial dimensions of these endosomes. And so, given an image like this, where we have fluorescent signals that are lipid, endosome, and DNA, now we can use the software I've developed. Again, I've built off of this landmark paper by Crawford Greer for locating the center of fluorescent spots. But we can basically decide which one of these are nanoparticles co-localized with a RAB versus nanoparticles that are not. And the results of such an assay are shown here. So what I'm showing are low and high membrane charge density, and the number of nanoparticles co-localized with RAB5 and the number that are not, at 2 time points. And again, we found sort of 2 unexpected findings which sort of confirmed here quantitatively. First of all, most of these nanoparticles are not co-localized with RAB5. Second of all, low and high membrane charge density don't show statistically significant difference in the number that are co-localized. This is surprising because the transfection efficiency of low and high is over 2 orders of magnitude. So we expected to see a massive difference in the amount of co-localization with these early endosomes, and we don't see that. So the wild type RAB5 here, it sort of, it showed us some interesting things that the RAB5 is actually used in the endosomal trafficking of these particles, but it also raised some interesting questions we couldn't answer. And to answer these questions, we turned to a mutant version of this RAB5, Q79L. So Q79L inhibits the GTP hydrolysis or slows it down, which results in a large number of fusion steps relative to recycling events. Now, phenotypically, what you'll see are large, giant early endosomes because this RAB5 cannot leave and the early endosome cannot mature in the late endosome, which is what happens in the wild type case. And so when we express this mutant RAB5 in cells and incubate them with nanoparticles, this is what we'll see. These large circles are individual early endosomes that have swollen to the size of microns so that they're actually, the lumen and membrane are distinguishable. And inside these giant early endosomes, we can resolve the individual nanoparticles. So with the wild type case, I couldn't really answer Pierre's question directly, how many nanoparticles are inside the endosome. But using this mutant, you can actually distinctly count in the individual nanoparticles inside each one. Now, what's interesting is that not only can you differentiate between nanoparticles that are in the lumen or in solution inside the endosome versus the ones that are bound. So now you really do have an assay for studying the interaction of these nanoparticles with the endosomal membrane. But even in the case of high membrane charge density, you'll see that nearly all the intracellular nanoparticles are actually co-localized with or found inside these early endosomes. And so now we can sort of explain those 2 unexpected findings from earlier. Most of the nanoparticles were not co-localized with early endosomes because early endosomes, in the wild type case, have a very short lifetime and quickly mature into late or recycling endosomes. And so most of those nanoparticles that we saw in the wild type case that were not co-localized with RAB5 are most likely in a late endosome or a recycling endosome. Now, we did not see a statistically significant difference between low and high membrane charge density because endosomal escape does not occur from the early endosome. If it did, we would have expected to see more of these nanoparticles outside of those early endosomes in the high membrane charge density case. So this wild type Rab5 raised these two interesting questions. We sort of answered these questions with the mutant Rab5, as well as developed an assay for measuring endosomal escape. Because now by slowing down the maturation, this ambiguity about lack of colocalization and whether it means it's outside versus inside is gone. Right? With the mutant case, when you're not colocalized, it means you've escaped that early. Now I want to emphasize that with the formulations I've shown here, we do measure transfection efficiency. So we do know that some of them eventually do escape the endosomal pathway. But the work with the wild type and mutant really ruled out escape from the early endosome as a possibility. So
is Rab5 absolutely necessary for the endosomal uptake to occur? Did you try a null mutant?
no, I did not try a null mutant, and there
What's
are
in the literature?
yeah. Yeah.
What does the literature say
About whether Rab5 is necessary.
for integrin-mediated uptake?
No.
Or uptake of these kinds of particles in general?
Yeah, no, so it's not necessary because you have dynein and clavin. Those are really the necessary proteins for at least these two internalization mechanisms. But my understanding is the accumulation of Rab5 at these sites is not necessary. It's necessary for the formation of this early endosome. But that's only based on what I've seen in the literature. I can't confirm it in my work. So sort of to summarize the three main parts of my talk, I showed you novel structures that form when we add peg lipids to the complexes, such as these thread-like bicycles. I've shown you that we really have two strategies for escaping endosomes: high membrane charge density and pH-sensitive peg lipids. But there's really two barriers when you pegylate your lipid DNA nanoparticles: internalization as well as endosomal escape. And finally, I've shown you that lipid nanoparticles traffic through early endosomes but escape at later stages of the endocytic pathway. So with that, I'd like to just point out that gene therapy suffers a lot of similar criticisms. It's fusion physics. And, they think it's never actually going to get done. We're always 10 years away, but there are clinical trials ongoing. Right? 5% of them use these lipofection vectors, and it's really going to be a combination of these small contributions to the field that move it forward. Right? So, this is what I've talked about today, what's shown in bold here. All the software is actually on the group website, and will be available in this book chapter for other people to do these types of measurements. And I've worked on other projects with postdocs and then projects and graduate students in Sirius's group. I did not talk about them today, but these are sort of the publications. And with that, really, I want to acknowledge everybody who was involved in this work, and most important is my PI, Sirius Athenia. He's really given me a lot of freedom with this project, but also provided me invaluable advice in terms of where to move next. And the other main people that have contributed to what I've shown today are Kai Ewert, shown here. He's the project scientist who synthesizes these peg lipids. And then Xiaoling and Bruno were two postdocs who I've worked extensively with, Xiaoling specifically for the hydrolyzable peg work. Bretton Fletcher, who's here, is an undergrad who sort of worked with me on numerous projects throughout his time here at UCSD. And I also want to thank the National Resource for Automated Molecular Microscopy, who were formerly at Scripps La Jolla and now at the Neurostructural Biology Center. They were really instrumental in doing that cryo-EM work. And of course, there's numerous people like Peter Chung and Emily and Victoria, the new graduate students in the group, as well as Yuli, who's here. None of this is really possible without their advice and input during group meetings, as well as help directly in the lab. I like to say, you've been in graduate school long enough when you need two pictures of a group to really. So a lot of these people, they actually couldn't be here today. Like Bruno and Xiaoling, they moved on, but they were instrumental. So with that, I'd like to take any more questions.
So the floor is completely open for more questions. Anyone?
I just had a stupid question about the cryo-EM of the thread-like micelles. How do you know that they're thread-like and not standing up lamella?
Yeah, that's a great question. And the reason we know that is because we're assuming there's peg lipid in them. And so that, because of the spacing in between them, and that peg lipid is gonna prefer that thread-like shape. Now, I do wanna say there are examples in that image of what could be lamella. Get back to it. And that it is actually perpendicular to the surface. By the way, that's not a very energetically favorable configuration. That's another reason we've sort of ruled that out, because this is a very thin water film. You wouldn't expect sort of such a short lamella there. But this gray region here could very well just be a flat tri-layer sheet. And I also want to emphasize, in thicker regions, you do see them doing what you mentioned, like they don't necessarily have to be parallel to the hole.
Now, regarding the heterogeneity in this picture, when you actually design a drug or a vehicle that takes a drug somewhere, the FDA wants it to be absolutely homogeneous.
Yes.
And of course reproducible. How do you get from this to a homogeneous?
Yeah. So what I'm showing here are lipid DNA nanoparticles that self-assemble by just mixing the components together. And that's why we're seeing this heterogeneous distribution. Now, if you wanted to have a monodispersed collection of these nanoparticles to really move ahead, you sort of have two options. One of them would be to use a column to separate based on size, so that you only have a collection of a single size. And another more common technique that's really picked up traction lately is to actually use a microfluidic device, which can sort of just produce perfectly homogeneous nanoparticles every time. Now, for our purposes, we actually preferred working with equilibrium nanoparticles because it's sort of a little easier from a modeling standpoint and understanding the interactions, not necessarily a kinetically trapped nanoparticle, which is what a microfluidic device would give you. But I do want to say also on top of that, that although liposomes can form such a heterogeneous distribution, Doxil, which is a pegylated liposomal formulation containing doxorubicin, is FDA approved, and I don't think every single Doxil particle has the exact same number of lipids. So when you have an assembly of molecules, you do have a little wiggle room.
So do you see any use for this elongated?
Well, I mean, working in the MRL, I can imagine a lot of uses actually in terms of possibly a mask for nanolithography application. People are trying to design smaller and smaller structures. So this is a very cheap and easy way to produce these uniform spacings. But in terms of drug delivery, I know there are people interested in worm-like structures for drug delivery, but with what I've done with nucleic acid, I can't imagine a direct use to deliver nucleic acid.
So first of all, congratulations, beautiful work, and I'm just so jealous when I did my PhD thesis, all I could do is a few light scattering and I was exhausted. So you do light scattering, you do focal microscopy, you do cryo-EM. So this cryo-EM, so this automated, I'm curious. So how does that work? Do you what is their part? What do you do? Now, so this picture here.
Yeah. So, I bring the sample to them and basically.
So you go there.
I go, yeah, I went
Okay.
to schools.
Okay.
Yeah, exactly. And they were nice enough to let me operate the microscope a few times by myself, but they are a little protective of the electron microscope. So it's not anyone can stroll in there and use it. And so, you prepare the sample by adding it to this, to that holy substrate I showed you earlier. It's plunged in liquid ethane using the Vitrobot. And then the most challenging part in the experiment is once it's vitrified in liquid ethane, it has to be transferred to the microscope in the presence of liquid nitrogen. So, I got to try my hand at that a few times, but I was very fortunate to have them with me sort of helping along with it. It's pretty tricky to deal with a sample that's constantly immersed in liquid nitrogen.
It has to be that cold?
Yeah, so, well, there's two things. It's plunged in liquid ethane so that you form an amorphous layer of ice here. You don't want the ice to crystallize. You want to really study these things in their native state. So crystallization of the ice will damage it. And once it gets that cold, if you were to pull it out and hold it up in the atmosphere,
Sure.
ice crystals from humidity would start to form.
Sure.
So you just have to keep it in liquid nitrogen the whole time.
What's the temperature of liquid ethane?
Liquid ethane, I think it's -180. But yeah.
And most importantly, the cooling rates are just incredible.
Yeah.
It's colder than liquid nitrogen, I just don't.
Yeah, well, it's not
Which
necessarily
the cooling rate
temperature,
is
it's the
important.
heat capacity
There's
that
a heat
makes it better than
capacity,
liquid
too.
nitrogen for the visual.
So on the second part, right, the transfection, the PEG RGD stuff, since the motivation is just for in vivo transfection, so when you do these cell studies, do you actually use serum? That part I missed.
Yeah. So, we found this formulation using MBL5 and GMO that works in the presence of serum, but everything I've shown today was done without serum. We've tried it in serum, and it's not that surprising. I mean, you see with the pegylated and RGD type stuff, and most lipid formulations, you just see a decrease in efficiency due to serum proteins binding on the surface of these particles.
So here's a teaching moment. Do you have a piece of advice for the graduate students that you leave behind?
I mean,
What would you have wished you had known in year 1 or year 2, or would you differently?
well, there's sort of the smaller details and then the bigger picture things. So I'll start with the big picture thing, which is, to do good science, it's really you have to find a perfect balance between trying different things and then finishing things all the way to completion. So don't get caught up in one project, but don't keep bouncing around looking for like the next greatest discovery. In terms of an individual thing that I wish I had sort of done from the beginning is, these Rab GTPases, I expressed them in cells transiently, so I delivered a plasmid so that Rab GFP would be expressed. I wish from day one I had started working on developing stable cell lines that have Rab GFPs, because it would have made the measurements a little less noisy, it would have made everything a little easier to understand. And generating a stable cell line, from scratch, it can take a while. So it's something I've been working on for a while now. You just need time. It's a time-consuming process. Also, siRNA, actually. I wish I'd done it. Plasmid DNA is not as hot as siRNA now, so I wish I had done the siRNA nanoparticles.
But the siRNA, all the companies are dumping their
Yeah.
siRNAs. siRNA is very hot in universities, but there are a lot of larger pharmaceuticals that drop.
It seems to me that antisense is actually going to beat it up, because you can make antisense molecules that are resistant to nucleases. You can't do that with siRNA because it has to be going into a physiological pathway. And you can actually, and this may do away with your the need of gene transfer. You can actually now upregulate proteins with antisense.
Yeah. So to explain to everybody else in the crowd, what I was delivering was a piece of plasmid which contains about 4,000 base pairs in a circular strand of DNA. Antisense are very small, short pieces of DNA. And now, typically, siRNA is also a short strand, but it binds to messenger RNA. Antisense has a completely different mechanism of action that occurs inside the nucleus. Also, from an endosomal escape point of view, getting a large plasmid out of this structure is much more difficult than just small antisense or siRNA molecules. I really expect to see a completely different dynamic of escaping the endosome if I had smaller nucleic acids.
Do you know what your limit is, size limit?
Size limit of what?
Of what you can transfer this from?
Oh, what the largest that can be transferred? So it was shown almost 15 years ago that you can deliver entire artificial chromosomes using lipid particles.
That's one advantage to viruses.
Yes. Yeah. So the viruses have a finite capsid size. These synthetic vectors you can design as large as you want.
I mean, this is peripheral, I just was curious about. So the acidification of the late stage endosomes?
Yes.
Those are proton pumps, they're pumping in protons?
Yes. So
And what turns them on? I mean, why aren't they, are they active before
Yeah.
endocytosis? Or-
Yeah. So what happens is, like this cartoon is pretty good for explaining it. You know, you have these different mechanisms of internalizing cargo inside cells,
Yeah.
and then they form this early endosome.
Okay.
And eventually what will happen is that, what's happening is that RAB5 is really giving this the early endosome identity because it recruits and interacts with other proteins that mediate its trafficking. Eventually, RAB5 will leave this early endosome and RAB7 will accumulate. And when this happens, now you have enzymes like hydrolases, which are delivered from the Golgi to this late endosome to break down the cargo. And it also, like you were saying, there's also proton pumps that require sort of RAB7 and its effectors to really get to the surface and do their job. And it's a very well-regulated process. In fact, some of the, like as an example, there's a membrane-bound receptor in the late, or a membrane-bound protein in the late endosome that delivers the hydrolases here. And RAB9 actually takes it back so that this receptor, this membrane-bound protein, doesn't get degraded in the lysosome. So you have a whole collection here using RAB9, I believe the other one is called AP1, that sort of regulate the composition of these late endosomes. But yeah, it's not a sudden drop in pH. It's a very slow, like the early endosome is slightly more acidic than the outside, the late endosome is slightly more acidic.
How acidic does it get? Like 5
Yeah, so
or what's
the early endosome can get down to as low as 5.5 pH. The late endosome will go down to about 5, and then the lysosome, I think, at its most acidic would be about 4.5. And it's also not just the acidity, right? I didn't really mention it during the talk. There's also all those other enzymes whose function is just to break down material.
Okay, let's thank Ramsey again, and then if you guys can leave.